Now I have to link those two videos together. Great. Unknown caller, you just cost me like 15 minutes of my life. And rest assured, it was probably just trying to sell me something. Lab's canceled, I'm furious, we're done. All right, plasmid DNA, purification, quantitation. Don't worry, we're going to talk about plasmids and what they are in more detail as we move forward. In the regular semester, this sort of falls into sequence a little bit differently. So the protocol references plasmids. They're important, all right? But they're not on your lab midterm, and we're going to talk about them in an upcoming lab, so no need to worry. You could remove the word plasmid and have a better idea of what today's lab is. Today is all about DNA purification and quantitation, which is just a different word for quantification, all right? So we're going to be using a kit, all right? A DNA extraction and purification kit called the Wizard Plus SV Mini Prep DNA Purification Kit, all right? If you work in a bio lab, you'll see a lot of these on shelves in the lab. Let me grab the actual kit as an example for you. All right, so this is not the same kit, but it's a similar one, and it's by the same company, Promega. All righty. So you'll see these, you go into some labs, they'll be on a big shelf. Sometimes they cost $400, sometimes they cost $1,200. What are they? It's a bunch of containers with special solutions in them. And usually it has a bunch of these tubes that can go into a centrifuge, or you can put the tubes into a thermal cycler, which gets them warm and then cools them off. See, just a bunch of containers of expensive liquid. All right, that's it. Sometimes they're proprietary components, sometimes they aren't. When they aren't, oftentimes labs will just make the solutions up themselves and then not use the kits. But the kits are like, they make it simpler because everything's pre-made. It's like a meal kit or something. It's like HelloFresh, but it's not all rotten inside there and overpriced. It is overpriced, quite frankly. I'm not a fan of HelloFresh. I use Cook Unity. Use code BIO105 for 10% off your first order and to show the channel some love. And while you're there, don't forget to like and subscribe. So we're going to be extracting some DNA out of some E. coli. Why would you want to do this? Maybe to sequence the thing. All right, there's lots of reasons you want to get the DNA out of a cell. We're using a special kit. This protocol is just rewritten version of the protocol that is inside one of these kits. Just that simple. So, if you're working in a bio lab, somebody might say, oh, I need you to extract DNA out of that sample over there. And you can be like, okay, I don't want to ask any questions. I'm just going to look in the kit, read the protocols they've got, and they'd probably give you all the clarification you need. Anyway. We're going to extract DNA, and after you extract DNA, you want to know two things, right? One, did I extract the DNA without any contamination? Because usually you get protein contamination if you don't do the steps right. And if you get protein contamination, then your sample just gets thrown out. It's no good. It's not pure enough. Okay? So one question we'll have is the DNA that we extracted pure enough? Do we have a pure enough sample to ship out for sequencing or to do anything with? And two, how much DNA did we get out? Okay? Because usually after a DNA extraction, you're sending the sample off to someplace else to be sequenced. If there's not enough DNA, or if the DNA is contaminated, then you just paid, shipping and to get the DNA sequenced, just to have them be like, hey, the DNA gave no good results, so you didn't do the purification correct. All right? This is I've got a friend who makes more than twice the amount of money I do by doing DNA extractions with this kit and then screening samples for DNA purity and quantity. And she hates her job because she does what we're going to do today over and over and over again with a PhD. But she lives in beautiful Salt Lake City. And she's from what I hear, very happy. She just got married. It's lovely. I got to go to the wedding. Her name is Dr. Jess Toth. She's a very nice person. I'm so tired. All right, let's just get into this. So how do we measure how pure our DNA extraction is and how much DNA we got? How do you know? That's crazy. We're just going to use another, even more expensive spectrophotometer called the nanodrop. And we're going to measure absorbance at 260 nanometers and 280 nanometers. Those are outside of the visible light spectrum. And DNA is invisible, as is a protein, But that's okay, because it still absorbs light, just not in the visible spectrum, okay? And the nanodrop can just deal with that. The nanodrop can also deal with very, very small quantities. So, yeah. Great. So, I'm just going to run you through the math part real quick, okay? We use absorbance at 260 nanometers to determine the quantity of DNA. We use the ratio of the absorbance at 260 to 280 to determine if there's contamination of the DNA, all right? A 260 to 280 ratio of about 1.7 to 2 is usually considered acceptable. Anything outside of that range indicates contamination. Now, if the number's higher, that indicates one type of contamination, and if it's lower, it indicates a different type of contamination, but that doesn't really matter for our purposes. If it's contaminated, it's contaminated. Question?
Yeah, sorry, did you say the 260 is for purity or for amount?
Amount.
Okay.
So the 260 is for quantitation, and the 260 to 280 ratio is to detect purity.
What's the okay level for the ratio?
1.7 to 2, all right? So if we go right here, a 260 to 280 ratio between 1.7 and 2 are pure enough for most molecular biology techniques. Sometimes it's a narrower range depending on what your technique is that you're using. Question?
Is there like I guess a generic standard curve for determining there's one with absorbance of the 260 and how that relates to like mass or like concentration? Is there like a direct formula for that?
Absolutely there is, and here it is right here. So I didn't talk to you about Beer's law, the Beer-Lambert law, but it can be really useful because technically you can use spectrophotometry to determine the concentration of something without a standard curve if you know a characteristic of the molecule called the extinction coefficient, which is just how quickly light gets absorbed as it passes through the sample. It's how quickly the light gets extinguished, right? It is. It's a very thick name, and the symbol is like one of those E's with the crazy loop-the-loop in the middle. So it's all cool, the extinction coefficient. So this equation here is basically just using the Beer-Lambert law and simplifying it, all right? The Beer-Lambert law, you also need to know the length that the light passes through. Extinction coefficients, you can look them up. There's like huge databases of them. They've all been determined, right? But you don't have to ever use the Beer-Lambert law here, all right? You'll use it if you take like a physio course, probably. Anyways, I got all distracted. Here's the rule. For every 1 unit of one absorbance at 260 nanometers, assuming the sample is pure enough, it's about 50 micrograms per milliliter of double-stranded DNA. So if I took a DNA extraction and I put it in the nanodrop and I measured an absorbance of 2, how much DNA would there be in there? 100 micrograms per milliliter. It's just that simple, folks. Check it out. Let's look at the table down below, and I'll show you. So if you took some samples, right, and you got these results right here, you would say, oh, sample A has contamination, disregard it entirely, right? Sample B, that's a very acceptable purity. Yes?
I'm sorry, how do you know?
Because the ratio of 260 to 280 is not between 1.7 and 2. A ratio of 260 to 280 of, I believe, like 1.8, that's exactly what pure protein should have. So if anything else is there, it will disrupt that ratio. Now, technically, right, I could have this table just have absorbance at 260 and absorbance at 280 as 2 separate columns and make you calculate this ratio. But no piece of equipment is not going to just report this for you. You know, like the nanodrop already just reports this ratio. So I don't want you to worry about having, you're never gonna have to take an extract sample and put it into a nanodrop and measure it at 260 and then measure it again at 280, because the nanodrop's an incredibly advanced spectrophotometer. It'll measure a full range and give you a whole curve and then calculate the stuff for you right in the machine. You'll see today when we do our samples. But here's the thing, folks. This sample, it's no good, so we don't care about this. We disregard the whole sample. If that happens, you go, damn it, I have to do another DNA extraction. I wasted 3 hours. And you just stay up until 4 a.m. And you just do that, and then you finish graduate school, hopefully. You know? Then you make other people do it. Okay, so this sample here, the ratio's good. The absorbance at 260 nanometers is 2.79. So to calculate the DNA concentration, all right, which here is in nanograms per microliter, but that's the same as micrograms per milliliter, believe it or not, because each of them is a difference of a factor of 1,000. All right. All you have to do is take 2.79 and multiply it by 50, and that gives you the concentration of DNA. Make sense? Sick? Sick? Yes, please.
Just wanted to understand the unit conversion. You say those are actually compatible, the same, like nanograms?
Yeah. Check it out, I'll show you. I'll show you with my favorite thing in this whole wide world, which is dimensional analysis. Which is dimensional analysis / conversion factors. All right? All right, cool. 139.3 nanograms per microliter. Fix that. Microliter. Okay? There are, and we wanted to get it into micrograms per milliliter, right? Because that's what these units are here. Oh, cool. So check this out. All right? There are 1,000 nanograms in every microgram. And there are 1,000 microliters in every milliliter. Both of these statements acceptable and true, right? So 1,000, 1,000, 1,000 divided by 1,000 is 1. Units cancel out, and it's the same. It is cool, right? And it never hurts to double check this stuff. You just write it out. And I love it when you can write it out and be like, I don't have to put anything into a calculator, you know, at all. I just cancel out units. Sick. Any questions there? So today, here's what you're going to do. You're going to run through this protocol right here. All right. Don't worry about the demonstration video. This was, I didn't edit that out from during, quarantine times. You're going to run through this protocol. You're going to do it bench by bench. Each bench is going to prepare 2 samples, sample A and sample B. You've got E. coli in the fume hood, okay? So you put 1.5 to 2 milliliters of the E. coli. Take it from separate E. coli cultures, all right? Because there's going to be multiple tubes back there. Each one's a different culture. Boom, pretty chill. You put it into a micro centrifuge tube, which you should have at your benches. No tubes. Oh, those are all the components of the kit, which Ashley was nice enough to actually partition out for you, so you don't have to go into the big box kit and pipette anything out. No, yes, you were right. I'm sorry, I said no to you and you were right. I give you 5 points. They're right. Why do I have the camera? I'm losing it, folks. Put this here, all right? These are the tubes. It's only Tuesday. But we'll be fine. So check out this box, right? It's got micro centrifuge tubes. You're gonna take 2 of them, label them A, label them B, put E. coli in, put these tubes into the centrifuge. It spins around. You get a pellet. Remember the pellet, the booger at the bottom? Yeah, that booger is gonna have only E. coli in it. So you'll have all the E. coli you need. You pour off the supernatant into the waste material. The supernatant is the fluid, all right? You pour it off into your waste material thing. You might have to give it a couple of shakes. You might have to give it a little tap. That booger is gonna be stuck down there pretty effectively, okay? Then you do it again, so you have lots of E. coli. Then you're going to add celery suspension solution, which is right here. Man, Ashley keeps knocking it out of the park. It's right here, labeled as CellRes for celery suspension solution. That's going to help you resuspend the cells. You resuspend the cells, all right? Then you have to break the cells open. You add cell lysis solution to split the cells. Nothing too crazy. Then you add alkaline protease solution. What do we know about alkaline protease solution? It's got an enzyme, it's alkaline, right? So it's basic, it's not acidic, right? And it's got a protease in it, all right? That's gonna help you break down those proteins, which are your contamination that you don't want. I'm gonna put this back here. Okay, we run through that. Then you add neutralization solution. It neutralizes what's going on. See, everything's pretty chill so far. It's all done in the same tube. You really want to make sure that you get the cells resuspended. I always forget to mention this. So if you're looking at your protocol right now, you want to highlight this part if you're going to be running the protocol. Is step 5, because resuspending by pipetting isn't fully explained. What I mean is, you just need to take your pipette, all right? Put a tip on it. There's no tip on this one, okay? There's gonna be that booger down in the bottom. You eject the air, not all the way down to the second stop, just to the first stop. Then you just put that pipette tip in there and you go up, down, like this. And you're basically blasting it with a little jet of water, which will get the pellet to resuspend. And that's usually good enough after a few rounds to get it to, break up. You've got to get that pellet to break apart, otherwise you won't be able to lyse all the cells. Okay, all of this is still happening in the same tubes. Add neutralization solution, deactivate the alkaline protease, because the alkaline protease is most active at alkaline pHs, right? So you just change the pH, you make the enzyme not work, you neutralize it. Pretty cool, right? Do not vortex, all right? That'll give you contamination. Centrifuge this again, okay, at step 9. And if this is these are points to make notes in your protocol. What's going to happen now is the protein will be in the juice, okay? And the pellet will be cellular debris. So you're gonna make these spin columns which the components are right over here. Technically, one piece is like the spin column and the other is the collection tube, but when you put them together, they're referring to them as spin columns here, okay? Put this in front of the camera just in case. There it is. It's got two components. Boop, boop. All right. This little tube, this big tube, they go together. At the bottom of this little tube is a piece of filter paper. So what you're going to do is take the liquid out of your tube, put it in here, and then centrifuge it. Woo, 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 woo. Just do it like that. Just spin in a circle. It'll work. Don't do that. Use a centrifuge. Okay? That's going to push the sample through the little piece of filter paper right here, right? And you're going to get all the DNA stuck on this filter paper. When you're running these protocols, here's the lesson I want you to get today. When you're running these protocols, the most important thing is, where is the thing I want? All right? So for most of this, the thing you want, well, it started in the E. coli, then it goes into the liquid, then you're going to run it through the spin column, and then it's going to be right here. Then you're going to do a process called column washing, where you just wash, wash, wash. You just push more liquids through this. The DNA is a charged molecule. It has a net negative charge. So it's going to stick to this polar, right, filter paper. All right? And you just keep washing it with nonpolar solutions. Those nonpolar solutions will pull off anything else that's not the DNA. That's the idea. So you do a couple of wash steps, okay? Wash, 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 wash it again, and then at the very end, you take the column out, which has your DNA, which should be relatively clean, on the spin column. You put it in to one of these, okay? And you put some water, which is a polar freaking molecule, people. Why is water so polar? Oxygen's so dang electronegative. That lets it serve as the terminal electron acceptor in the, electron transport chain, right? Everything fits together. So at the end step, you're going to take some water, nuclease-free water. Nuclease-free water is free of enzymes called nucleases, which degrade DNA. We are covered in nucleases. So if you want your sample to stay, intact, gloves, all right? Why are we covered in nucleases? Because if you've got DNA on you or in you that's not inside a nucleus, you don't want it. It's probably viral DNA, right? Because your DNA should all be inside of a nucleus. All right? The last step, take this, add some water, centrifuge. That water is going to pull the DNA right off of the filter paper. This step is called eluting. All right, you're eluting the DNA, you're pulling it into suspension. And then this tube will have some water and DNA in it. Once you're there, let's go to the back. I'll show you our nano drop. It's cool. We're going to use the one that I like. Good, the new ones. During fall and spring, they bring down the fancy new one. It has its own screen on it, and it's just dumb. I hate it. Ours in the back room works better, and it runs through a computer, so you don't have to worry about the internal operating system becoming out of date or having to pay them for a license, and they can't just brick your piece of equipment, which they will 100% do if you work in a research lab. And you try to not keep paying the company, $1,500 a year. They'll be like, well, you can't have the updated software, and we made the old one obsolete, so your piece of equipment doesn't work anymore. It's a wild world we live in. Yeah, question.
At the beginning, we have two tubes, A and B. Are we doing separate containers of E. coli for each tube?
Yes, so you want to use a different culture for tube A and tube B. There are, I believe, 4 cultures in the fume hood at the back. It's just to give you a replicate, so you're more, in case One of the cultures didn't work right. Or maybe the bacteria grew poorly, yeah.
We're effectively doing everything twice.
Yes.
Or like
well, and it's
steps simultaneously.
simultaneously, yep. And it's gonna make your life easier because it'll make the centrifuge balanced. Working with one tube, you would still have to repeat everything, but just with a balancing tube. So it's really just as easy to do 2 tubes. Plus, regularly I see with benches, like one tube just works better than the other. You know, sometimes life you just get a bad one. Okay, that's it, folks. Any questions? Did people get notes about those key steps? Just always double check, like, where's the DNA? And if you want to know, ask me. Keep track of which step you're on as you work through this. This is all you. Enjoy, have fun. It's going to be a hoot. We got plenty of time. This whole process takes about an hour usually, so. Sick. Oh, and does anybody need a protocol? Got one extra protocol. Wow. Yes, we got a taker. Perfect, it was meant to be. All right. Oh, thanks. Ooh, let's 3-hole punch it. Let's do both. Why not? It's free. Just do all the services. It's like adding 8 creamers to your coffee, you know? Like, oh, here you go. Here, you pass it on.
Oh, thank you.
All right, we stopped the recording.